challenge that we are trying to tackle here is a huge one: making vaccines more effective. I'm Raquel Cuella Martin, and I am an assistant professor in functional genomics at the Human Genetics Department. During my PhD, I had done a few small experiments, but it was really on my postdoc that I started a project working with genome engineering technologies, like right there, state of the art, and trying to making them work at a larger scale. Having been a pure molecular biologist and having encountered some of the challenges that we have in the lab, and then seeing the power of those technologies, I was like, all right, this is a whole new way of doing the molecular biology, so I should just move those technologies to my day-to-day research. It doesn't matter if it's a cancer vaccine or if it's a vaccine for an infectious disease. We really need to harness the power of the T cells. The research project just takes those T cells that express a specific receptor that will recognize or will bind to that viral particle or will bind to that tumor protein and trigger an immune response. We go with our genome engineering tools and make mutations on that receptor. So we modify, and then we measure, okay, when we modify here, does it recognize better? Does it recognize worse? Going in a very systematic manner and trying to understand how we can make modifications that will make that more specific, more efficient binding, more efficient immune responses for better vaccine generation. I would say the project as of now is really platform building so that we can get the tools to generate those data sets. And that is something that we can even achieve in the lifetime of this D2R project. The true inspiration behind this project came when my colleague, Heather Mellicher, and I really got together, had discussions here and there from her immunology perspective and also from my genome engineering perspective, how we can make something that actually works, that is actually scalable, and that actually will lead to meaningful biological discoveries on how the T cells recognize the foreign peptides, the foreign antigens, the foreign viral particle or cancer cell. I really think it's a really nice example for the true collaborative efforts that that are happening in D2R. Equity, diversity, and inclusion considerations were coming up at the very beginning. The T cell receptor recognizes this little antigen coming from a virus or from a cancer cell that is presented by another receptor that is called the human leukocyte antigen. And that receptor, we call HLA, is the most different across humans. It's highly polymorphic and it varies also very largely across populations. When you make a vaccine or when you make an adoptive T cell therapy, you do want it to work a bit more broadly. From the beginning of the project, we thought, all right, we need to test TCR, antigen, HLA combinations that are not only specific to white overstudied populations, but also in underrepresented groups. We chose that list for our proof of concept studies based on picking those T cell receptor antigen HLA pairs that could be present in African American populations or in Hispanic populations. This project fits very well with D2R, ultimate D2R mission of delivering effective RNA vaccines or RNA therapeutics. Those therapeutics, the way they work, they need to elicit a robust, durable immune response. And that is largely dependent on the T cells. So how we design those vaccines, how we design those pieces that we put on them is critical for that response to happen. And for that, we really need to have the understanding that we are trying to gain with this platform here.